Can you see my first slide, please?
Yes, we can.
Screen is visible? Acha. Good evening, all of you. Namaskar. First of all, I'm grateful to Dr. Mansi and Bharati for giving me an opportunity to share with you some of our work which we have been doing in the NCL, and as well as in my previous institution I had done it. So all of you know NCL, so this is nothing new, but this is a U-shaped building. So I am on the west side, and the second floor, my office and lab is located, in case some of you would like to visit anytime NCL. So before I start with my talk, I would like to introduce my scientific mentors, those who have helped me to come up to this position. See, I did my PhD from one of the CSIR lab at Lucknow, which is known as Central Institute of Medicinal and Aromatic Plants. And Lucknow is the only city in India which has got 4 CSIR institutes. Even Bangalore is known as a scientific city, but it doesn't have as many as scientific institutions like Lucknow. So I did my PhD at CMAP in 1998 with Dr. R.P. Sharma, and then afterwards I got selected for the DAAD Fellowship of Germany, and there I went to a big carbohydrate chemist. He's the father of considered as father of carbohydrate chemistry. So I worked with Professor Richard R. Schmidt at the University of Constance. So many of you may not know that in city of Constance. So I just briefly let you know that Constance is the southernmost tip of Germany. So if we compare with our Indian map, then it is almost like Rameshwaram. So the half of the city is in Germany, half is in Switzerland, and it is surrounded by a big lake which is called Bodensee, and that covers 3 countries: Switzerland, Germany, and Austria. So there I worked with the Professor Schmidt on the glucosyl transferase inhibitors. So this is the this is the foyer of NCL. All of you have those who have visited. Sorry, I think this is okay. So this is our founder director had written in the foyer of the laboratory that the purpose of this laboratory is to advance knowledge and apply chemical science for the good of people. So when every morning when we enter NCL, so we have a motto what we are supposed to do and how we can help our society. So these are my current research interests. Actually, basically, I have interest in natural product chemistry. So natural, I will later on I will introduce what is natural product chemistry. So when we do natural product chemistry, I have interest in anti-malarials and anti-cancer, and TB also we used to do, but now I do not have a collaborator who can do the anti-TB assays. Since I worked with Professor Schmidt and on the enzyme inhibitors, so I have interest on protease, and then we do also total synthesis of small bioactive natural products based on carbohydrate scaffolds, because this is my contribution or this is towards Professor Schmidt's that whatever I learned on carbohydrate chemistry with him. So when we say we do natural product chemistry at CSA or NCL, so our idea is that we do curiosity or target driven isolation. Then whatever molecules we isolate from natural products means plants, microbial cultures, animals, like this. And so then whatever compounds we isolate, we do undertake structure elucidation of those molecules. Then we try to find out the biological activity of those molecules so that it could be used further. And if they are biologically active, then we go for mode of action. And since sometimes the compound plant may not be a good source of the compound. It may give us the molecule, but we cannot harness in future. So we do undertake the synthesis of those molecules, and also the molecules whatever we isolate, we do the chemical transformations, structural transformations, in order to increase the either biological activity or solubilities. These issues we try to solve through the chemical transformations. And we are lucky that we are in the one of the located in the Western Ghats of India. So I would slowly take you towards the how the natural anti-malarials from nature were discovered initially. And before doing that, I will show you two slides, which I am sure many people may not be knowing that these are the natural products, but we have been using in our day-to-day life. And one of since the audience is in a diverse background, so and I'm an organic chemist, so there will be some structures in my slides, and you have to please bear with me because we cannot work without structures. So I will be showing those structures, but I won't be going in the details. So this is the fantastic molecule which is called aspirin and acetyl salicylic acid, which we have been using from so many years and in different form, either aspirin or dispirin, or those who have heart problems, they regularly take ecoaspirin. So this is aspirin is actually a natural product which nature had given us. Now it is synthesized, but the actual molecule was given by the nature, and it used to be found in the willow bark. The analgesic effects of willow juice are described in even before 1550 BC, and that this juice was applied by the father of medicine Hippocrates and Pliny an elder for the reduction in the for the use as an analgesic. This thing, the isolation of aspirin molecule was done in 1897 by the German scientist called Felix Hoffmann, and he was working at Bayer AG in Wuppertal, Germany. See, this is the molecule, which is the drug molecule, which has within a short span of time has come into the market, because most of the drugs they take at least 7 to 8 years minimum, maximum could be 15, 20, 30 years. But this is the shortest path taken by this molecule to from the discovery to the drug as a for clinical use. So it was introduced in the market by 1899 by Bayer again, and this comes under NSAID. And basically, as I told you, we all know that it is for treatment of pain, inflammation, and fever, and also and is used as an anti-clotting agent for the prevention of heart attacks. The best known and most widely used medicine in the world, 100 billion aspirin pills weighing 400,000 tons are swallowed yearly on Earth as well as in the space stations. And this is the only inspiring molecule which was see. This is there are many drugs which are developed, come into the market, but they get phased out. But this is the molecule which was developed in early 19th century, but still being used. That is the beauty of this molecule and the role of natural products. So now I will be discussing with you the how what the work we have done with the malaria. But before that, I would just simply or I would like to introduce malaria. Generally, when we think of malaria, we think it's a useless disease and all these things. But trust me, this is a very severe disease, and if it is not treated in proper on time, then a person can lose his life. So malaria is the most common of the parasitic diseases in tropical and subtropical regions. As you can see that the world map, it is all red, that those are the endemic countries or the region in the world, and mostly 40% of the world population is under the malaria. And malaria has been not a problem of of this modern age, but it has been in a problem in India for centuries, and it has been even encrypted even in Charak Samhita. And Hippocrates referred this around 2500 years ago. The second photograph, Dr. Mansi, is it possible to get a pointer in this zoom?
I don't know, Ashish ji, how
I
to
was
do that.
checking, but I could not see it.
Pointer is
Where is
there in
it?
the Zoom. You can check in the
How to
Zoom.
get
There
it?
is a pointer.
Just a minute. Yeah, yeah. Okay, I got it. Just a second. Huh? So, can you see the arrow now, which I'm moving?
Yes, we can see.
Yes, it is visible.
Okay. So, this is the second photograph. If you see, it is the most famous photograph. This photograph is of Alexander the Great, and it is believed that he died at the very early age, at very young age, at the age of 32. And it happened when he conquered India, when he was going back to Europe. On the way, he died. And it is believed that he died due to malaria, which he had contacted in India. And if he had been alive, say the map of Europe would have been completely different. It's a global epidemic, and it is a called, as I mentioned, this is a caused by a parasite which is of the genus Plasmodium. But in humans, there are four species which are Plasmodium falciparum, vivax, malaria, and ovale, which are the cause for disease, which causes disease in humans. And out of this, falciparum is the most deadliest because if it is not treated, the most of the deaths occur in humans due to the attack of Plasmodium falciparum. And this human body, when parasites enter, it infects the red blood cells, and it is, we all know that it is the due to the female Anopheles mosquitoes. This parasite is transmitted. Basically, symptoms of malaria include fever, headache, and vomiting, but and it usually appear before between 10 and 15 days after the mosquito bite. And in India, this is the problem of several. Many times physicians are not able to diagnose malaria based on symptoms, mostly in the eastern part of India, like in Bihar, some parts of West Bengal, and eastern part of UP, where Kala Azar is very randomly available. Pandemic is there, so the initial symptoms are similar unless the blood pathology is done. So symptoms, many times physician treat for Kala Azar, and then the situation becomes very difficult, and almost the patients, they lose their life. And if it, as I was mentioning, this is if it's not treated, malaria can quickly become life-threatening because it disrupts the blood supply to vital organs. And many parts of the world, these parasites have developed resistance to a number of malaria medicines, which has aggravated the more treatment situation. See, before this onset of COVID, I'm very sorry to say that malaria was the number one killer disease. Now it has been it was surpassed by COVID, and it is generally in 219 million worldwide people, they get contact malaria, and out of that 660,000 deaths occur each year, and unfortunately children below 5 years of age are the victims. And highest burden countries are 68% of where 68% of malaria deaths globally occur in the 10 highest burden countries. And who are at risk? As children below 5 years, pregnant women, HIV infected pregnant women, people with HIV/AIDS because they have the low immunity. International travelers from non-endemic areas like in Europe, it is not now the malaria is not there. But when they travel to Southeast Asian countries or Africa or South Asia, South America, they do get they contact malaria and then they take back to their respective country. And also the immigrants from the endemic areas they take it and then they spread it. Malaria has one interesting India connection. See, this is Sir Britain's Sir Ronald Ross. He was working in the Presidency General Hospital in Calcutta, who finally proved in 1898 that malaria is transmitted by mosquitoes. Before that, people knew that intermittent fever comes and people die due to fever, high fever, but they were not knowing that it is the disease is due to the mosquitoes. And he did this by showing that certain mosquito species transmit malaria to birds. And he isolated the malaria parasite from the salivary glands of mosquitoes that had fed on infected birds. And for this work Professor Dr. Ross received the 1902 Nobel Prize in Medicine. Is one of the early Nobel Prizes he got it and was awarded to malaria work only. And in 1998, Government of India commemorated the 100 years of this discovery by releasing a postal stamp in honor of Sir Ronald Ross. So this is the malaria life cycle. On my left hand side, this is the malaria parasites, how that goes, how it replicates, and the cycle goes on inside the mosquitoes. And this right hand side is the human, malaria parasite when it enters a humans, and then how this goes in. So briefly, I will tell you. This is it goes into the when the mosquito bites a healthy human, the malaria parasite first goes to liver cell and then it infects the liver cell and she joints are formed and then this rupture takes place and this she joints then enter the human blood stage. So there are two stages: human liver stage and human blood stage. And these immature trophozoites they form the ring stage, and then mature trophozoites again she joints are formed, they rupture and again go to the blood cycle. And this is the stage when the parasite enters the human blood stage, then the patient develops the symptoms. And this Plasmodium vivax, ovale, and malaria, they work on the gameto. They are they develop in the gametocyte stage. So this was all about the malaria infection and how that life cycle goes in, and the natural products. The when we say natural product molecules to medicine lies not only in their pharmacological or chemotherapeutic effects, but also in their role as template molecules for the production of new drug substances. So not only the many times natural product itself is not used, but it it influences and it gives us the ideas to the organic chemists to develop molecules based on the natural product, and that those are being used in the for the treatment of various diseases. The example is the aspirin itself. So, what are our treasures? We have more than 250,000 species of higher plants, and many of which are under threat of extinction by the next century. And in Ayurveda or in our traditional system of medicines, almost approximately 10,000 species have only been used, and many species have not been put to systematic chemical and pharmacological examination. So this is the structure of quinine. This is the first anti-malarial which was isolated from the natural sources. So its natural source is Cinchona officinalis, and this is a Peruvian plant. And from the bark of this tree, quinine was isolated. And it was first isolated by Pierre Pelletier and Joseph Coventry in 1820, and its molecular formula was given by a German chemist Adolf Strecker in 1854. And generally, it is given as a 3 milligram per kg of the body weight. So, 2 for 200 milligram tablet is for adults. And if you see on the right hand side, this is the coat of arms of the Peruvian government, and it had this Cinchona officinalis had so much importance in the Peruvian society that the tree has place in the coat of arms. And this was a highly guarded tree. These Peruvians, they did not allow anyone to take this out of from their country, although the French tried several times, and later on Spanish people, they succeeded, and they brought it to other their colonies in the Americas. So as I was telling, that sometimes natural product itself is used, or it gives an inspiration to synthesize some other molecules. So quinine was the molecule naturally occurring, and but afterwards, it also parasites also developed a resistance against the quinine. So then synthetic chemists, they came up with the new molecules based on the template of the natural product. So it was the first was chloroquine in 1934. Germans had synthesized it. Then came primaquine, sontoquine, pamaquine, mefloquine, and mepaclin, and so all. But all these quinine-based molecules, parasites have very efficiently developed resistance, and these are more or less non-effective. In Chinese system of medicines, what the Chinese have been used, they do drink a lot of tea of many herbs and flowers. If you see any Chinese, they will keep on drinking. They don't drink water. They will be drinking jasmine tea most of the time. If you go to any Chinese restaurant, also they do serve jasmine tea. So Artemisia annua is the plant. It's actually weed, and it has been used in traditional Chinese system of medicine as a cure for malaria over 2000 years. But in 100 years back, nobody knew that this is malaria. I mean, disease was known as malaria. So people had intermittent fever. So whenever they will get fever, high fever, intermittent, so they would be using a decoction of this tea, this leaves, and so this fever will be cured. And since all the effect anti-malarials available at the time in 19 during the Vietnam War, the already parasite had developed a resistance, and what happened? Lot of American soldiers, Vietnamese and Chinese, they were dying not due to bullet, but they were dying due to the malaria prevalent in Vietnam. So a big program was launched by US government also, and also by the simultaneously by the Chinese government, to find out effective anti-malaria so that soldiers' death is controlled. So what they did in 1970s, the Chinese government started a program. So they took the plants which had already shown some activity against fever. Since already Artemisia annua was being used traditionally, so what they had taken out this plant, and found that Artemisinin, which is in Mandarin dialect known as Qinghaozhou, was the active constituent in the plant, and the Chinese scientists isolated in 1972 and showed that this is the molecule due to which fever is reduced, and malaria could be a ideal molecule for treatment of malaria. So this is actually Professor Yo-Yo too. She was in the Shanghai University in 1972. She started this program, and she too. This is the plant of the Artemisia annua, and she isolated. This is the structure of artemisinin, and this artemisinin, she showed that not only she isolated this molecule, found out the structure, but she also said that out of the whole plant, only this molecule is the active molecule for the treatment of malaria. And for this work, she was bestowed with 2015 Nobel Prize in Physiology or Medicine. So there are three major secondary metabolites of Artemisia annua. One is artemisinin, which is the anti-malarial compound, and it has in the plant for biosynthetic pathways are like that. Artemisinin is synthesized from artemisinic acid. This is the biogenetic precursor for artemisinin. And if you see the chiral centers, these two chiral centers, the C1 and C12, these are same in both these three molecules. And artemisinic acid is considered as the biogenetic precursors. So plant synthesizes artemisinin from artemisinic acid, and artemisinic acid is itself is synthesized from artemisinin B. And artemisinic acid and artemisinin B are biologically inactive molecules. They don't show any antimalarial activity. Only artemisinin is the antimalarial compound. So this artemisinin is very unique molecule. What this has actually, I will take only a few seconds to describe it, or just this structure. This has 5 oxygens. There are many molecules which have lots of oxygen, like anti-cancer compound Taxol. It has around 8 oxygens. But when you look at the crystal structure of this molecule, all the 5 oxygens are on the one side. And this example, I think you are able to see my arrow. So this 1, 2, 3, this is called 1,2,4 trioxane, and this 1,2,4 trioxane moiety is the reactive group, which causes makes it anti-malarial. If you remove any of this oxygen, it is no more anti-malarial. And these are the two Plasmodium falciparum, Plasmodium parasite strains. One is W2 Indochina and D6 is Sierra Leone. Sierra Leone is a country in Africa notorious for all the antimalarials develop parasites develop resistance against antimalarials, and these are the mostly reported from Sierra Leone itself. And as you can see, this artemisinin is very active. It's 2.93 nanogram per milligram per ml. And all that, as I mentioned, all the oxygen atoms are on the same side of the structure, giving it a unique structural features. So it is there are a lot of uniqueness in this molecule, which chemistry wise we know. So I won't be
Definitely.
discussing. Yeah, sorry,
Also, there was
sorry, I could not get any questions there.
No, Ashish, maybe due to some.
Okay, should
Yes.
I go ahead?
Yeah, go ahead.
Okay. So, as I will just briefly describe this. As Artemisinin showed structures of activity, so many chemists they come up with so what we can take out from the molecule or what we can add to increase the activity. So, this is one of the molecule. This is deoxyartemisinin. One oxygen is removed, but because of that, it is no more antimalarial activity, but shows very weak anti-cancer activity. If this ketone is taken out and make a CS2 is a deoxyartemisinin, so this is 12 times more active than artemisinin, but highly toxic, so it could not become drug. So when we reduce the ketone to artemisinin to dihydroartemisinin, then active antimalarial activity remains intact, and it is the precursor for synthesizing many artemisinin-based drugs, which I will be showing in my subsequent slides. Sorry. So this is the difference between quinine-based molecules and artemisinin. So artemisinin acts at the red blood stage with the erythrocytic stage, and this palmaquin is attached at the hepatic stage. So this is not very, this is very deep, because how artemisinin works in the how kills the parasite. So this is the C4 radical. This Gary Kushner and Paul O'Neill, they proved it. They showed that this is the C4 radical, and this C4 radical makes the hemozyme and causes the death of the parasite. As I said, artemisinin was isolated from the plant, so many synthetic organic chemists took up a challenge that we can be replace the plant and we synthesize it in the lab. So several total synthesis of artemisinin have been reported, but all these methods are not suitable for the commercial production because almost 17 to 20 steps are there and minus 78 many steps you have to do my reactions at minus 78. So these are not industrially feasible. So therefore, Artemisia annua remains the only source of the production of artemisinin on a large scale, and Artemisia annua was introduced and cultivated in India by CIMAP in 1985. And this is a Chinese plant, but I must tell you that the germplasm is not China. So CIMAP did not get the germplasm from China. The founder director of CIMAP, Dr. Akhtar Hussain, he visited Kew Laboratories, but Kew Botanical Garden in UK. From there, he had procured the seeds, and these seeds and CIMAP had many regional centers, but one was in Pulwama, Srinagar. So Kashmir was ideally, has more or less same weather like China, where it grows, and so it was introduced there, and slowly this plant has been introduced in the plains, northern plains, and now it grows even in Maharashtra, Gujarat, Rajasthan, Madhya Pradesh, and IPCA is the largest producer of artemisinin-based drugs in the world, and they have a world WHO contract. So all the artemisinin-based drugs which are being supplied to the world, African continent, so they are the sole proprietor there. So they are producing artemisinin through the contract farming. So this is the work which we had done at CMAP Lucknow, and we had developed several technologies for the production of this artemisinin, and we had patented this. We generated a lot of IP around this, and we had sold to three companies. We had sold our technology and patents to these three companies, and Themis is making this email 3 ampules for the treatment of malaria. So since, as I told you, many natural products, they are active, but sometimes they could not become drug because of some of the inherent problems. So artemisinin also is not spared. So it is also has some problem, like it is poor soluble in oil and water. So this is a major problem because how to administer the drug, and short plasma half life. That means it goes in the body, so it's short is the half life is 30 minutes. So within 30 minutes it is excreted out of the body, so then it is not able to clear the parasitemia because recurrent, and then due to this recurrence of malaria happens, and poor oral activity because it is not water soluble. So modification of artemisinin have been done. So make it oil soluble, arti-ether, artemether. So we had the patents there, which we sold to Themis and Morpen and Cadila. So this is the production of arti-ether, and also sodium artesunate, artelinate, which are water soluble. So when we say it is oil soluble, so these are mixed in the groundnut oil and given intramuscular, whereas sodium artesunate or artelinate can be given orally because these are water soluble. So I'll just briefly tell you how the artemether is, the oil soluble derivatives or water soluble derivatives are prepared. Artemisinin is reduced to dihydroartemisinin, and then it undergoes etherification to prepare, say, to give us arti-ether or artemether. Arti-ether is ethyl derivative, artemether is methyl derivative, and instead of etherification, if we do esterification, we will get the water soluble derivative like artesunate or artelinate. And as I mentioned to you that artemisinin, in the one of the previous slides, I showed you that artemisinin and dihydroartemisinin have similar activities. There is no decrease in the biological activity if you reduce this ketone lactone to lactone. So artemisinin to dihydroartemisinin is the same. And when we reduce this and make ether, then we have two stereomers, diastereomers. So one is beta is solid and alpha is oil. So and similarly, these are also water soluble derivatives. With this is solid, but this is also water soluble, so they become, because sodium salt, so it becomes solid only. And their activities profile, if you see, this is same. So based on this, there was a problem because in the DGCI had approved our technology based on 70-30 ratio of beta to alpha, and this ratio in during synthesis maintenance of this ratio is sometimes difficult. So what we thought at NCL that we will make stereoselectively beta ether derivative only. It is solid, so then we don't need to do any further purification. So it will reduce lot of carbon footprint, carbon fingerprint, and we will save some lot of electricity and chemicals because silica gel is used and that goes waste. And so we developed a method for the stereoselective synthesis of beta ether from dihydroartemisinin. So I won't go much in the details. And we had patented it and we have published in Asian Journal of Organic Chemistry. So our work was published as a cover page article in Asian Journal of Organic Chemistry. And this is all of you know. This is the Lakshman Jhula and this is the Swagla. And why we chose this is a single catenary bridge, and our peroxide linkage in artemisinin is also like a bridge endoperoxide, so this is handy. So we thought it ideally resembles the Lakshman Jhula, and the editor was very impressed with this cover art, so they published as a cover page article. So everything is not good with artemisinin because now it is being used since 80s or worldwide in many parts of the country world. These derivatives are being used. So there are chances that parasites will also develop resistance against artemisinin. And unfortunately, after artemisinin, there is no other antimalarial which is even in clinical trials. So if the parasite develops resistance against this artemisinin, so we do not have anything to fall back. So that's why in India, artemisinin is not given at the first dose. So what DGCI has developed is it's a SCT, artemisinin combination therapy. So it is a with already known antimalarials like primaquine, mefloquine. So in between, in a combination with these other molecules, other antimalarials, the artemisinin is given so that widespread resistance does not develop. So, but in 2005, Jambu et al. In Lancet, they reported that some of the patients which were treated with artemisinin derivatives had shown susceptibility against some of the plasmodium resistance. And they isolated from 530 patients, and they showed that there is some susceptibility towards developed by the falciparum, and these patients were from three countries, and these three countries are very notorious as far as resistance in malaria is concerned. This is Cambodia, Thai Cambodia border, French Guiana, and Senegal. So most of the chloroquine derivatives or quinine derivatives resistance has been reported from these three countries only. And so now the artemisinin resistant strain has been spreading from Southeast Asia to other parts of the world, but it's not very much, but still some sporadic cases are there. So what we need, because there are no other molecule, so we need to think beyond artemisinin. So we have to look for new naturally occurring antimalarial agents, ideally directed against new biological targets. Then this happened, in 2009. I was invited by United Nations to develop a plain lecture in Trieste, Italy. So it was a workshop on malaria. So one of my friend, I mean not friend, I didn't know him at that time. One scientist from ICGEB, he had come over there. So he works on malaria biology. So I work on the chemistry of antimalarials. So what happens? So there is a one enzyme which is called falciparum, and this enzyme is, I won't go in much detail because these are different classes. So it's the cysteine protease falciparum 2. This is the enzyme which is available in the food vacuole of parasite, malarial parasite, and the role of this falciparum is to degrade hemoglobin to amino acids in smaller units, so that those smaller units can be taken up by the virus, by parasite, for its own growth and sustenance. And if so, what we thought, that if we can have some molecule which we can inhibit this falcipain too, I mean it doesn't allow to work this, then the parasite will not be able to degrade the hemoglobin, and it will die of starvation, and there will not be any replication. So in India, they have this falcipain. This ICGP scientist, he had this. They have this falcipain enzyme. So they were doing the biology part of it. There was no drug discovery. So he's of my age. So one day evening we were moving around. So we said, "You are also working on malaria. I am also working. Why not? We joined together and we developed some new antimalarial drugs." So we started at that point of time. So I thought that we will have dual acting hybrid molecules. Hybrid because we are having two molecules, but both the molecules will lose their identity and become a new entity. So that is called hybrid molecule. So artemisinin will be already is known for clearing parasitemia, and falcipain 2 inhibitor is required. We will introduce in the artemisinin, and so that will block the falcipain enzyme. So the parasite will die because of starvation, will not grow much further, and artemisinin will clear the parasite from the human blood stage. So what we thought, that we will have an artemisinin structure, have a linker, and we will have a molecule which will be uh, identified by the enzyme. So it will go and bind with the enzyme, rendering it ineffective. So it has to have a proper falcipain, because the X-ray structure of falcipain enzyme is reported. So we know that there are two sites, P1 and P2 pocket. So P2 pocket you have to put a heavy molecule, and P1 you have to put a electrophilic site, so that which will go and bind with the falcipain. So based on this, we had designed almost more than 10 compounds, and we were very lucky that 3 of the compounds were found to be the most effective. As you can see, these are the number. I won't discuss much these structures, but these compounds are, against falcipain 2 are 5.9 micromolar. But if you see its EC50 values, it is 2.7 nanomolar, 3.3, 2.57. So actually we got a molecule which is 10, 15 times more active than the artemisinin itself. But it works again on a different target. So it is different than artemisinin. So even if the resistance is developed against artemisinin, eventually it will happen. So we will so it will not be this parasites will not be developing resistance to this molecule, because their mode of action is different. And in vitro cytotoxicity also we had carried out. These were not found cytotoxic. So these molecules are unique in that way, that it can be taken up further. And parasitemia clearance from the body was from the in the mice were 100% almost. So, and not only this, we had tested this molecule, our molecule against, we got from US artemisinin resistant strains of Plasmodium falciparum, and against that also we tested, and our compounds were very effective. So yeah, so this is the again inhibition of this same data, but we had synthesized because one site was a diastomeric mixture, so we made the chiral molecule and found out which chiral molecule is active. So R is only active, S is not active. So just to in a nutshell, if we want to say that we got 3 molecules which shows picomolar range activity against Plasmodium falciparum, and these compounds effectively inhibit survival of ring stage parasite. Hybrid molecule showed complete parasite clearance in vivo using Plasmodium burgei, because Plasmodium burgei is given in the mice for the in vivo test. And mode of action is inhibited falcipain 2 and block the hemozoil formation. So by that way, it became effective antimalarials. So based on this work was very interesting. So we are in right now we are working on making it a water soluble, because so that it can be given orally. So we are trying to make the water soluble derivatives, and we are in discussion with pharma company. So take it for phase 1 or further clinical trials, because those are very expensive things, and we are academicians. We can't do those work at our place. It requires completes a different set of expertise. So this work was we patented it. So we have European patent, US patent, Chinese. We didn't go for all countries, because wherever it is endemic, we will go. We went there like China, India, Brazil. So in China, it has in January only it was granted our patent. So and we published it in the European Journal of European Journal of Medicinal Chemistry, and this work caught the attention of Times of India. So our work was highlighted in the front page of Times of India national issue on 23rd August 2020. So I'm coming back to this acknowledgement, because this all work was done by my students. So at present, Balaji is doing Balaji and Ajay are involved in Artemisinin work. This is Dr. K.C. Rana. He was my first PhD student. With him, I had started this work, and now he's in Vadodara working at Syngenta. So Ishwar did some work. Sayantan did some other work which you have not covered here, because that is also artemisinin synthesis we did from other route. And so he's now in Cipla, is a group leader there. And this Lakshmi Goswami, she just graduated, so now she is going for a postdoctoral research in Technikon, Israel. So my collaborators, I'm grateful to them, the Asif Mohammed and Pawan Malhotra, because they have the falcipain 2 enzyme, and they have been doing this biology work on our molecules. So we have been funded by DBT, CACR, DST, SERP, and I still get funding from DAAD. So they haven't forgotten us. So I get a book grant every year. I get a €200 book grant, so I can order any scientific book. With this, I thank you all for your patient hearing.